Hello, this is Dr. Krishnagowdu Saravanan, Assistant Professor, Postgraduate and Research Department of Biotechnology, Arignar College, Arts and Science, Krishnagiri, Tamil Nadu, India. I'm very much happy to present this slide. The title, Recent Advancements in Soybean, that's called as in glycine. From this glycine marks, this is an important oilseed legume in the world which produces tofu, soy oil, soy milk, soy sauce, and various soybean related products. Soybean meal is a byproduct of the soybean oil which is extracted from the uh, waste process. This mainly used for the poultry industry and some other purposes. Coming, these and all various products which is available in the market for the better nutrition purposes. Coming to the world oil production, US, India is in leading countries for the production of oil. The soybean market is projected around 185 million metric tons to 260 million metric tons in 2026-2028. This slide, which represents that soybean nutritional values, which possess a number of nutrients, mainly protein, fat, carbohydrate, dietary fibers, minerals, vitamins. This is in world soybean production statistics. Brazil is the leading state for the production of soybean, around 37%, followed by US 31%, then Argentina, China, and India. India is the fifth most largest soybean production country around the world. In India, Maharashtra, Madhya Pradesh is the leading state for the production of soybean, followed by the Maharashtra, followed by Rajasthan, Gujarat, and other states. Why we need to improvise the biofortification in the crops? The second global conference on the biofortification, which already conducted, and then the the famous scientist M.S. Swaminathan spoke about the biofortification and the zero hunger challenges anchor the goal achieving by the 2025. This is the year 2025. We cannot fulfill the biofortification approach. Third biofortification conference was held in 2017 in India. At the time, 400 plus various scientists from across the globe were attended this meeting and then spoke about the biofortification approach. Coming to the world statistics of the biofortified crops, around 150 biofortified varieties of the 10 crops are already raised in 30 countries. Remaining 12 crops, which is supposed to be released in the in upcoming years around in the 25 countries. In India, the first biofortified crops is named Swagam. The commercial name of the Swagam is called as Prabhani Shakti, which is released by the ICRISAT Hyderabad on 5th July 2018 through the conventional breeding of the improved iron and then zinc biofortification process. Why we need to improvise the biofortification approach? We need to reduce the hidden hunger, vitamin deficiency, maternal health, and ammonia and child mortality. This complete biofortification not only improves the nutritional benefits of the particular peoples, but indirectly impact on the agriculture by offering various nutritional element and minerals which cope up with the biotic and then abiotic stress tolerance and also improvise the yield. That's why we are in my research we are chosen the flavanol biosynthetic pathway. In that flavanol biosynthetic pathway, Arabidopsis thaliana MYB12 transcription factor were cloned into PKMA1301. This particular biosynthetic pathway were classified into the cyclic pathway like biosynthetic pathway from phenylalanine, cinnamide, forcamide CoA, chalcone, flavanone, dihydroflavanone, then flavonols like quercetin and then rutin. This is quercetin and then rutin chemical structure where the various uh, health benefits activity which is available in this particular compound, mainly anti-cancer, antioxidant, anti-malarial, anti-diabetic, in a number of anti-inflammatory and then various properties have. This is the major biosynthetic pathway of the flavonols. Why? What is the major impact on the salinity of the particular countries? This is overall Indian scenario of the salinity stress, which is affected in the Indian country. In Tamil Nadu, around 3.68 lakh hectare of the lands has been degraded by the salt condition. Um, in topmost state is called as a Gujarat. Around 22.22 lakh hectares of the lands has been contaminated with the salt, followed by the Uttar Pradesh, then West Bengal, and, and other countries. In India, around 6.7 million hectares are currently facing the high accumulation of the salt, and which leads to the 20 million hectares by 2050. What is the major negative impact on the salt stress? Salt is a major stress which negatively significant to the yield and reduce the plant growth and the development, imbalanced iron homeostasis, decreased pigment production, increased ROS and then RNS, and then high expression of the genes. We can clearly emphasize as this graphical abstracts which represent the soybean which around 55% of the yield loss followed by the others. Maize is around 60% of the yield loss. Coming to the MYB12, why we need to choose the MYB12 for the biofortification approach? This particular MYB12 which belongs to the MYB R2 R3 MYB transcription factor which contains the MRE MYB recognition domain and followed by the recognition sequence also. Three structural independent MYB class families were identified, namely 11, 12, and 111. These three are the flavonol biosynthetic specific activators in flavonol biosynthetic pathway. This particular MYB12, which targets only two structural genes, mainly CHS and then FLS. CHS means chalcone synthase and FLS means flavonol synthase. That's why the pathway which turn into the biosynthetic production of the flavonols alone, which inactivates the DFR. DFR means dihydroflavone reductase. This is the key enzyme for the expression of the anthocyanin content in the particular pathway. That's why we have chosen that ATMYB12 over expression for the increased production of the flavanol content in soybean. This is the normal transformation protocol. We need to check the parameters like how much transformation efficiency is that. That's why we're able to check that agrobacterium related transformation in 1301 vector, PKMBA 1301 vector, which possesses hygromycin phosphotransferase 2 as an plant selection marker, GUS as a reporter gene. From this, we can able to incorporate into some physical kind of parameters of sonication and then vacuum infiltration. These two physical techniques which improvise the soybean transformation, we can clearly see that sonication 10 minutes and then vacuum infiltration in 10 minutes improves the soybean transformation percentage is in around 38%. We can clearly emphasize that the soybean seeds to after various stages like imbibed seeds, modified seeds, primary primary shoot regeneration, elongation, rooting, and then hardening processes. This is the image where we can clearly emphasize that GUS gene integration into the transformed lines of the immature leaf, mature leaves, flowers, and then part of the transgenic plant. After that, we have confirmed by using HPT2 presence in the transgenic lines. The gel images also show that presence of the band which indicates the 500 base pairs. This is overall key abstracts where the soybean were collected from Indian Institute of Soybean Research, Indore, Madhya Pradesh, India. Then the seeds were self-fertilized and then off-seed explants were prepared. Then simultaneously, the complete agrobacterium transformation where happens in 10 minutes of sonogation, vacuum infiltration 10 minutes. Then the seeds were co-cultivated. Then after that, the explants were washed with spetaxim for the removal of the agrobacterium contamination. Then the explants were inoculated into the primary shoot regeneration medium, in vitro rooting of the primary shoots, hardening. Then a complete process where takes place 22 months. This is the highlight of this particular key paper where we published in mm, very reputed journals called as 3 Biotech. The impact factor is in 2.6. And also, we have got that Best Oral Presentation Award in International Conference on Plant Biotechnology and Genome Editing, which is held in Kakatiya University, Warangal, Telangana, India, from 27th June to 29th June 2023. Coming to why soybean, why we need to incorporate the flavonol content. Soybean, already we discussed about soybean, globally economically important crop which possesses a rich amount of protein, nutritional values. For this purpose, we need to incorporate the flavonols in the particular plant, means we can improve the nutritional content through genetic engineering approach to eradicate the malnutrition content by eat edible soybean seeds. That's why we were framing some kind of experiments. This is the results where the cloning of ATMP12 in PKMB1301 by using specific enzymes SAC1 and then KPN1 restriction enzymes. This is a linear map where the KPN1 and then SAC1 were the restriction enzymes, where both the 5' and 3' the transcription vector were located. Then GUS as the reporter gene, HPT2 for the plant selection marker. This is a confirmationary image where the GUS and then presence of the ATMVP12. To eradicate the native MVB12 amplification, we have fixed the primers from the CAMV promoter of the external gene and along with the ATMVP12 for the reverse. That we got that clear emphasized bands of the transgenic lines. This is the HPLC results where we can clearly identify, see that non-transformed seeds showing around 57.7 microgram per gram of dry weight of the routine and then 33.3 microgram per gram of the routine content were analyzed. For in transgenic lines, the ranges varies from, we have analyzed around 7 different lines. The ranges from 87.09 microgram to 92.28 microgram per gram of the dry weight of the routine were detected, and then this fold increment around 1.5 fold increment was observed, and then quercetin from 42.2 percentage to 44.1 microgram per gram of the dry weight were detected. This is also showing that around 1.27 fold increment in transgenic lines. This is a chromatographical image where the quercetin and then routine were detected in the HPLC. This is a quantitative RT-PCR. That's like RT-PCR analysis of the seeds from the various stages like immature, mature, and then entire seeds after fertilization of the process. That's why we are designated R4, R5, R6. The entire pathways like PAL, C4H4Cl, CHS, CHI, F3H, FLS were highly upregulated in various biosynthetic pathway from the immature, mature, and then seed content. In this graphical abstract, we can clearly explains about that MYB12 overexpression improves the flavonol content by the incorporation with the agrobacterium mediated genetic transformation in the soybean. We can clearly emphasizes that various biosynthetic pathway regulating genes, mainly PAL, C4H, 4Cl, CHS, CHI, F3H, FLS were upregulated. From this upregulated means we can clearly see that quercetin and the routine content was upregulated, the total antioxidant content was upregulated, and then total phenolic and then flavonoid contents also upregulated in the particular seeds. This is the core objectives where the nutritional improvement of the soybean takes place through the biofortification of the flavonols, along with the protein and the nutritional of the particular already present in the soybean. This is an add-ons of the. Coming to the summary, we have developed the soybean transgenic line with improved transformed and then improved various uh, levels of the quercetin and then routine, and then a single crop with extensive nutritional property. After that, we need to screen the transgenic seeds with the salinity level. From this, clearly we can see that figure number C showed that reduced physiological and then morphological parameters because the salt content isn't more. From this, we can clearly the right side the image number D and then E is in transgenic lines with cope up with the salinity stress from 100 and then 200 millimolar of salt stress respectively, we can clearly emphasize where the transgenic soybean were connected to the MYP transcription factor were highly upregulated the salinity stress. We can clearly observe these images from this. From this, we can clearly emphasize that ATMYP12 not only improvises the nutritional content but also improvises the various parameters like salinity stress tolerance, to reduce the LPO content, to improvise the antioxidant properties, to improve the flavonoid biosynthetic pathway, and along with the improved salinity regulating genes and then proline biosynthetic pathway. From this, we have got the best poster presentation award in international national conference, which happens in National Institute of Biotic Stress Management, Raipur, Chhattisgarh, India, and also we have published more than papers which belongs to the uh, Scopus and the Web of Science Index papers. Um, 3 biotech with impact factor of 2.6, and then plant physiology and biochemistry with impact factor of 6.1, and then Journal of Plant Science and the Journal of Soil Science and Plant Nutrition with an impact factor of 3.4. And also I have awarded various awards like Young Scientist Award, Young Agricultural Scholar Award, DST Inspire Award from the Government of India, Indian government, and also we have submitted that one Indian patent, and then we have submitted 4 Indian copyrights. I have published 2 books, 15 book chapters, 14 publications, and 7 NCBA publications in GenBank.